Okay, good afternoon, everyone. Now, in the last class, I gave an introduction about translation, and we also discussed some things about genetic code. And that is all for towards building towards the chapter on translation, right? So, translation is something actually a very important lesson for several reasons. As I was describing in the last class, it has two things. First is it's the way the sequence information that is in the nucleic acids can become information in the form of proteins, and that is an important process. Say, for example, you have some text in English, and you are supposed to say translate into Tamil. It's actually a very difficult process because you have to think of many things. And translation, as such, is in literal meanings too. It is actually a difficult process. We have to know what the author, the original author, might have meant, and then convey the same intended meaning or effect or emotion. In translation in biology that we discuss, it is in nucleic acids, it's in sequence of nucleic acids, and that will result in or change it into language of amino acids, which will form owing to the number, composition, and the sequence. Then they will form a three-dimensional structure, and they may have several types of functions as proteins. So in amino acid sequence information, we have three things: number of amino acids, composition of amino acids, what type of amino acids are there and what are not there, and so on. And the next one is sequence of the amino acids. Basically, composition and sequence will also, if I said sequence of amino acids, then number and composition are also implied. But it's a good way to think of what makes information, what are all the constituents of information. So number and composition, if you know, it's not enough. What we should know is the sequence. Okay, in this lesson, we will learn, in this particular class, we will try to learn about the components of involved in translation. And in the later classes, we will describe about the mechanisms of translation. The focus majority of the time is on prokaryotes. And at places we will also discuss about eukaryotes, and there is one more about the regulation of translation and so on. The regulation part we will also probably discuss in the last unit, but we may touch upon it a little bit so that we'll have some basic idea. But otherwise, or in this class, we will talk about the components. When we talk about components, it's mRNA, tRNA, the ribosomes, and how the mechanism happens, mechanism of charging of tRNAs and so on. So I'll skip this. Yeah. So here we discussed about the genetic code in the last class, right? There are 64 codons. And out of those 64 codons, codon is a triplet. And the 64, out of the 64, there are sense codons, and there are 3 nonsense codons or stop codons. Right? And in the sense codons, one is specifically for start codon, and then there are other sense codons that code for all the amino acids, other amino acids. So we have 64 codons, and each consisting of 3 bases, and we only have 20 amino acids. And as I was implying or suggesting many a times, I want you to remember AUG, start codon, and the stop codons UAA, UAG, and UGA. These are the three stop codons. That's what you should remember. And in open reading frame, we also saw this picture. There can be the open reading frames. The codons are present where you do not call these three bases as a codon. Codon makes sense only when in the light of open reading frame. And in the context of open reading frame, now these three bases are one codon. This is second, third, and fourth. We can nowhere refer to these three bases as one codon, right? The triplet that is in consecutive triplets that start with a start codon and end with a stop codon is an open reading frame. And all those between the start till the stop, they're all codons. So open reading frames can be overlapping. We also discussed about the 6 reading frames, right? If you had a double-stranded one, reading frame 1, you'll count, or reading frame 2, or reading frame 3. The same way, we also have a reading frame for the bottom strand. So there are 6 reading frames. So you should try to know, have a look at it, try to feel, or try to engage yourselves trying to find out the open reading frames. Then we also, as I said, that is when you also take into account the opposing or the complementary strand, then you might be able to find out more open reading frames. Typically, you should remember that the open reading frame is represented something like this with an arrow mark. This would be a start codon, and this would have been the stop. And typically, you will have a promoter here. Okay, this would be +1, and you will have a terminator here. That's how something like that is representative of terminator. So when expression happens, that is transcription happens, mRNA is produced from here till here. So we also discussed about the reading frame, operon type organization in bacteria. In viruses, the most typical one is the the overlapping open reading frames. And what is special about eukaryotic, they all have, majority of them have very large introns. And there are numerous introns in most of the genes, right? You can remember viruses, majority of them have overlapping. Not that it's not overlapping genes are not present in others. It could be, but it's not as prevalent as you would see in viruses. Viruses, high gene density. Bacteria, majority of them are organized as operons. What is an operon? One promoter, and you have multiple ORFs and a terminator. So during transcription, it will produce one continuous mRNA, each with the open reading frame, and they will be translated to produce proteins. So this is basically the same thing what I have discussed in the last class based on the test. Codon, it's 3-letter code. They are read continuously. There are no gaps. Gaps in the sense there is no one base and you would skip that. That's not how it is going to be. Codon should be read continuously. And the start codon, as I was telling, there will be some exceptions. In majority of the cases, it is AUG in prokaryotes and eukaryotes. In prokaryotes, there may be some variations in some cases. It's not very prevalent, but people have identified that GUG or UUG can also be used in certain conditions. But typically, AUG is in majority of the cases, AUG is the start codon. Stop codon is universal, UGA, UAG, and UAA. So what is a cistron? It is almost something like an open reading frame, or you can also call it as a gene sometimes. So there is no clear-cut demarcation. Cistron is a DNA sequence encoding a single polypeptide chain that's equivalent to open reading frame. Or if it is not producing a protein, ORF, when we talk about ORF, it is about to give rise to a protein. But what about genes that give rise to just RNA? Right? That's why we use the word gene here. Right? Cistron is equal to gene. Gene may produce a protein, or may encode a protein, or may encode an RNA. I hope you remember what is monocistronic and polycistronic. In bacteria, they are polycistronic. That means there are multiple open reading frames like this. In eukaryotes, they are monocistronic, meaning that they typically produce one cistron produces one protein. One mRNA produces one protein, other than alternative splicing, RNA editing, and other cases. So there are additional, one part of mRNA is being able to produce or encode these proteins, something like this. Here you're shown a protein. Sorry, this is an open reading frame. It has 3 different open reading frames, start and stop there, and you have another start and stop and another start and stop. And each of them would be translated to produce a protein. And how is protein produced? It is by ribosomes. How do ribosomes recognize why don't they start here, or why don't they start translation here? It is dependent on something called as RBS, or ribosome binding site. So majority of the times the ORF is upstream of the ORF, you have the ribosome binding site like this. Okay, those are the regions where ribosomes will bind and then start translating from the start codon and will end at stop codon. Termination of translation occurs at stop codon. So ribosome binding sites in prokaryotes, they are called as Shine-Dalgarno sequence after the scientists who identified them, or simply they were also called as SD sequence, Shine-Dalgarno sequences. In eukaryotes too, there are certain ribosome binding sites with some sequence specificity. Those are called as Kozak sequence. So in eukaryotes, we will also discuss a few things more. Right, the 5' end has the triphosphate. That's the first base during that happens during the transcription. Whereas in eukaryotes, if you remember, we have some processing that occurs in the 5' tip of the mRNA in eukaryotes. So we have a G cap, right? G, P, P, and then you have whatever bases will follow. I hope you remember the structure of it. It's an important question that might come. And if you know that, if you know the G cap, and then one can assume that you know a lot of chemistry about the nucleic acids. Okay, so I think you should also remember the 5' linkage in the G cap. Okay, so the ribosomes have rRNAs. In prokaryotes, say for example, you have 16S rRNA. We will read about these in the later, but the small subunit of the ribosome has 16S rRNA. That 16S rRNA has a certain sequence, CCU-CC. This is from 5' to 5'. So I should have read it from CCU-CC. And there is also the ribosome binding site, the sequence is GGA-GG. It's not absolutely same in all the cases, but there will be variations to it, like the consensus sequence. We learned about consensus when we studied about transcription promoter elements, -10 and -35. Almost on the same similar basis, you can think of there is a consensus sequence that is the ribosome binding site. These are present somewhere 6 to 8 bases between from the start codon to the So this is the start codon here. It will be read this way, and there is a spacer 6 to 9, or it could vary a little bit. The spacer is present, and then you have some sequence like this, GGAGG, which is complementary to the sequence CCUCC, right? So by using the 16S rRNA sequence, the ribosome is able to bind to the ribosome binding site here. And then, so the spacer is about 3 to 9 bases here, start codon, and then translation occurs and it will be terminated here. You have another ribosome binding site, so a new ribosome can bind here and translate this region. And again, termination will happen. New initiation would have happened here, and so on. That is how 3 proteins would be produced from each, one from each open reading frame. So there is also, in a way, kind of the Cossack sequence is also similar, and the recognition of the mRNA by ribosomes is mostly dependent on this 5' cap. That's why it's very important for us to know the chemistry of it as well as its importance in translation. So the next is about tRNA, that is for transfer. T is for transfer RNA, and these are the key elements. Actually, when you talk about translation, our focus often is on the mRNA and the ribosomes. But I think tRNA is something that's even more important than other other components, because they are going to use, sorry, it is the central molecule for reading in nucleic acid. Say for example, this is the mRNA, okay, and this is, say, the say roughly a tRNA, and this is the amino acids, say for example. tRNA is the one that allows the translation of the, in terms of information, okay, information that is present in nucleic acids to that of amino acids and produce a protein. So they are the adapters for translating this genetic information. They're typically about 75 to 95 nucleotides. There are many types of tRNAs, and each type of tRNA can bind to only one specific amino acids, right? I think we should remember always these numbers. We have 20 amino acids. There are 64 codons, and out of which 361 are actually the sense codons. 3 are stop codons. You don't have tRNAs for it. So you can have many types of tRNAs. The least required, to our understanding, is about 20 tRNAs. Okay, we'll get to that in a while, but there are exceptions also again. So each type of tRNA is specific for only one amino acid, and you have to also, most tRNAs, this is a nice way of remembering when we read about genetic code. If we come across it, you would also remember that most tRNAs recognize more than one codon by the use of their anticodons. So it is, if you can see it here, this is the codon, and they have their the base 3 bases that are going to base pair with the codon is called as the anticodon. We'll come to that in the next slides. And here is the amino acid. So some of the tRNAs can recognize more than one type of codon. Again, this comes in light of degeneracy of the genetic code. I think we will discuss that again at some other point. But right now, you remember these things. Then the 3' end of the tRNA usually has a sequence, and that is the CCA sequence. That is where the site, that is where the amino acid is attached by, that is
Sir, we can't hear you, sir.
Yes, can you hear me?
Yes.
Okay, thanks. Okay, I think I was speaking about the modified ones. Yes, in tRNA, some of the important things that you have to remember is about the pseudo, some of the modified bases. We will discuss that in a while now. So, the structure of tRNA and also the processing of tRNA, there are some things that you have to remember. As a primary transcript that is just produced by transcription, you will find that the sequence is something like this. And then it is processed by two different enzymes called as RNase-D. RNase implies that it is an enzyme that cleaves RNA, but it's sequence specific, and these are specialized ones. So it cuts at RNase-D, for example, cuts in the 3' side of the tRNA, and RNase-P cuts on the 5' side. So these parts are eliminated from the structure of tRNA. And the bases, if you look at all the bases, they are normal bases, A, U, G, C. You would not find anything abnormal. And based on the sequences, they form arms like things, the hairpin loops like this one, and so on. This is the anticodon loop, and you will have other loops as well. We'll see it in the next slide. But then there are other modifications. First thing you have to know is the base modification. In the sense, several of these bases are modified. M is for methyl. Guanine is methylated in that region. All the methyl guanines, several of them, methyl cytosine is here, and you have a thymine here. This is pseudouridine, methylated adenine, and so on. Again, this is a general scheme. There are different types of tRNAs, and different types of tRNAs can have different modifications. Okay. And eventually there is the second one, as we discussed, there is 5'. The cut is made, 5' cleavage, that is by RNase-P, and 3' cleavage by the RNase-D. And then there is one more that is called CCA addition. That is this sequence here. CCA is added to the 3' side. So you should never read it, don't read it this way. That is wrong. ACC is wrong. You have to read from 5' to 3'. So CCA is the region. And this is 3' OH is very important because that is where the amino acid is going to be added. And then there is splicing, in the sense the yellow highlighted one here, yellow marked in yellow, the sequence is cut off, and the hairpin is somewhat made something like this. Okay. So that is the processing. It is produced as a complete, say something, we'll call it as primary transcript. Then you have several modifications, and finally we'll get a mature tRNA. This is specific for the tRNA that is for tyrosine. So the modifications, I think you have to remember a few things, if not all, and they are actually not very difficult. They are simple. At least with uridine, it is you can remember two bases. One is called as pseudouridine, and the other one is called as dihydrouridine. Say uridine, you know, the uracil, basically uracil along with ribose is the nucleoside. So you say it as uridine. Uridine triphosphate, if you said, then it would have meant UTP, right? Okay. So if the glycosidic bond is usually present on the carbon, nitrogen on one, on in position 1 of uracil, and it is bound to 1' carbon. Okay, this is 1, 2, 3, 4, and you would have a 5th carbon here. So in pseudouridine, the glycosidic bond is in a different location. It is present, it is bound to the fifth position, the carbon that is present in the fifth position. The chemically, if you look at it, it has the same composition, or same composition as uridine. There is only one major difference, that is where the glycosidic bond forms, right? So atypical of many nucleosides that we know of, or nucleosides that we know in all of nucleic acids, you see this is a different one. That's why it is called as pseudouridine. And the same, another modification of uridine is something like this: dihydro. That means two extra hydrogens to uridine. So those are present in this place, where the fifth carbon and sixth carbon would have had. If you look at here, they have the double bond there. They are completely saturated now by addition of two hydrogen atoms. So there is no longer the double bond. So you call it as dihydrouridine. So you will have many others, many other things. I don't need you to mug up all those things, but try to associate wherever possible with the what is in the name and how it would have altered the structure. If you know uridine, pseudouridine is different, is simple, because all you need to change is the glycosidic bond from position 1 to position 5. Dihydrouridine, you have only one double bond, and you add double bond between fifth and sixth carbons within uridine, and you add two hydrogens there, and then it is dihydrouridine. So there is the structural aspects of tRNA are very important, in the sense they play the major role. And if we understand tRNA completely, we would understand all of translation. So the structure, we know the processing has happened. RNase P cut here, RNase D has cut here, and the CCA enzyme has added this part on the 3' end. And there is processing and many things. Now we have the structure is, it's referred to as clover leaf model and so on. In at least in two-dimensional, you can call it as, this is D loop, because one of the conserved dihydrouridine is present in one of the loops here. You call the other one pseudouridine, a pseudouridine loop, because you have a pseudouridine base, modified base here. And this is where the amino acid is going to add. So you call that the acceptor arm. And there is one more, this one, that is where the anticodon is present. Anticodon is a 3 base, which will base pair with the codon that is present on the mRNA. So we call that the anticodon loop. So there is another loop that is called as variable loop. And in some mRNAs it's not present, some tRNAs it is not present, and in some RNAs it is short, and some RNAs it is some tRNAs it is longer. So it's kind of variable, so you call that as variable region. So otherwise you have to remember D loop, pseudouridine loop, anticodon loop, acceptor arm, and the variable loop. So if you look at it in the 3-dimensional, it's in 2-dimensional, it's easy to understand and see, and 3-dimensional it could become much more difficult to view, but it is worth trying to see those as well. If we are watching a GIF or something, other formats where it is rotated, I'll try to find maybe next class, and then you will be able to understand it better. And how does tRNA get charged with amino acids? What is meant by charging here? Charging means it is the binding of amino acid to the tRNAs. So there are two aspects to this. First thing is we should know the chemistry of how it happens, along with, and then the second one is the enzymes. Which enzymes and how do they do it? How do they accomplish this adding specifically to that tRNA, that amino acid? So in chemistry wise, you can think of it as there are two steps to it. That is called as addition of amino acid to the tRNA is called as charging. Remember that. So if there is a tRNA just like that, you would call it just say tRNA. But if it had an amino acid attached to it, then you call it as charged tRNA. So the first thing is activation of amino acid. Say this is the amino acid here. You have the CO carboxylic group. This is the amino group, and this is the R group. That is where the variable different amino acids will have different R groups. So the first thing that happens is the COH is going to bind with the alpha phosphate of ATP. This is ATP, adenine, triphosphate, alpha, beta, and gamma. So because as a result of which, this is the amino acid here is now bound to this AMP. It's a kind of intermediate that forms. And pyrophosphate, beta, and gamma phosphates are released out. So you call this as adenylated amino acid. If you want to say, here is something, if you want to say adenylated, that means addition of adenine base, only adenine. If you're saying adenylation, you're talking about the nucleoside, addition of a nucleotide. Basically, this is base, sugar, and a phosphate. So it's a nucleotide based on adenine. So you're saying it as adenylation of amino acids. So this amino acid is adenylated. That is the first part. It's kind of activating or energizing the amino acid. And then the 3' OH, where we had this sequence, the acceptor arm of the 3' OH, CCA, and you have the 3' OH. That 3' OH is going to bind to the carbon of the carboxylic group in the amino acid, thereby releasing the AMP. AMP is released, and then now the amino acid is bound to through a high energy bond. It is bound to tRNA. This tRNA is now charged tRNA. So I will quickly say a few things here. I hope you understood what is the difference between adenylation and adenylation. Only adenine base, then you call it as adenylation. Adenylation is the addition of AMP, adenine adenosine phosphate. So I hope you remember what is you understand what is charged and uncharged. This is the tRNA that you have here. This is uncharged because it is still not yet bound by an amino acid covalently. And this is a charged tRNA because it is bound by an amino acid with a high energy bond. There are some more variations in this. There are class 1 and class 2. Say we are talking about the tRNA. When we talk about the tRNA, we say it as 3' OH. If the 3' OH, if the amino acid is going to get bound to the 3' OH of this, the 3 terminus adenine, then it is class 2 tRNA. But there are also other types where the 2' OH of the ribose in that location. So you remember the structure of ribose, CH2OH, and then you have, this is the, say, a base, and this is 1', 2', 3', 4', and 5'. And typically, majority of the polymerization, everything happens is the 3' OH is typically involved. But in some case, the amino acid in general, as is shown in the class 2, the amino acid could be bound here, and that is class 2 tRNAs. In some other cases, you may have the 2', the amino acid could be bound to the 2' OH, and then those are called as class 1 tRNAs. Okay, so either way, it could be, once it is bound to 2' OH, it is also possible that the amino acid could shift to the 3' OH. So there is a kind of equilibration between them. But I hope you understood what is class 1 and class 2. If it binds to the first binding of the amino acid to the tRNA is on the 2' OH, then you call it as class 1. If it binds to class 2, 3' OH, then you call it as class 2. So here is the there are 20 amino acids, and then 20 amino acids, 64 codons. Then we need for the enzyme part, the charging of tRNA. We have seen the chemistry, and we need some enzymes that actually mediate this binding of amino acid to the tRNA, and that is done by enzymes called as amino acyl tRNA synthetases. Right, amino acyl, you're adding these to the tRNA. They are generating these amino acylated or charged tRNA, so you call them as synthetases. So there are 20 amino acids, 64 codons, out of which we'll only consider 61. Then how many synthetases should be there? That's a question. So there are there are variations among different organisms about how many synthetases should be there, or even how many tRNAs should be there also. So first thing is iso-accepting tRNAs. That means one synthetase can only accept one tRNA, right? So synthetase is something like this. You can think of, just for visualization purpose, you think of it like this. So assume this is the place for tRNA, and this is the place for the addition of the amino acid. So the amino acid should be attached to the tRNA somewhere here. So the tRNA synthetase should be able to interact with the tRNA so that only the correct tRNA comes here. And the synthetase should also interact with the amino acid correctly so that only the correct amino acid will come here, right? Otherwise, there are different types of tRNAs, and there are 20 different types of amino acids. So there is some degree of specificity required. Otherwise, the sequence is not going to be read properly, or even if it is read, the amino acids are going to be jumbled. So one synthetase can bind to only one type of amino acid, right? So the minimum number of synthetases you can expect is 20. There are alterations, we'll get to that. In the tRNAs, there are a lot of differences, and there are a lot of conserved sequences. So the tRNA synthetase will typically recognize the conserved sequences of that tRNA, and it will always be specific to the amino acid. Say if it is valine, for example, it will only add valine to this one. It will not be able to bind to other molecules. Okay. So something like this, if it is written in this case, there is a nomenclature glue tRNA GLN. That means the prefix says this is what is the amino acid that is attached to the tRNA. And the superscript, this one, indicates the codon that the tRNA recognizes. So I will try to, we will discuss this again too, just in case. So you have the mRNA and it has codons. And the tRNA has, I'm just representing only the antisense codon. Okay. The others you should assume they are there, the arms. So assume that this is 5' to 3'. So this should be 5' of the tRNA, and this should be 3' because base pairing is always anti-parallel. So here you would expect the amino acid to be there, right? Amino acid is attached always to the 3'. The charging of tRNA happens on the 3' side, near where the CCA sequence is present. So what is it interacting? What type of codon it is reading? Okay, that is what you put here. The superscript is about the codon it reads. And what you use as prefix here, glue, is the amino acid that has been added to it. Don't worry, it is confusing. It would be easier if I said, if I have written GLN for glutamine, tRNA and for GLN, right? That's simple. tRNA that recognizes codons of glutamine, and it is attached or charged with glutamine. That makes sense, right? But this is a little bit confusing. That is for something to understand here. In some bacteria, there is no glutamine tRNA. There is no specific tRNA for glutamine. And as I said, there are variations. We don't have to go into all, but it's a good way to see it so that we will remember that there could be variations to it. I'll just explain this and I will stop. Maybe I'll repeat in the next one. So there is only a single species of amino acid tRNA synthetase. So that tRNA synthetase would recognize, it recognizes both GLN and tRNA with glutamic acid also. So you can think of it as, sorry, if I said tRNA, no. In some bacteria, there is no glutamine tRNA synthetase. tRNA is present, but not glutamine tRNA synthetase. So there is a specific one synthetase is present, and that synthetase, say for example, I will put it as this. This is the synthetase. It recognizes tRNA that is specific for glutamine and also for glutamic acid. There are differences in the sequences of the tRNA. That is how you can tell them apart. And their anticodon loop will also be different, right? Because they have different codons. So what it does is this particular amino acid tRNA synthetase, it recognizes both tRNAs, tRNA for glutamic acid and tRNA for glutamine, and it will add glutamic acid. So assume that this tRNA that is shown here, it is added with glutamic acid, and it is glutamic acid tRNA attached that reads glutamic acid. That's fine. This would be a trouble one, isn't it? Because you have added glutamic acid, but it is a tRNA for glutamine, right? What is the difference between glutamic acid and glutamine? Glutamic acid it's an acidic amino acid. It has COH and then amino group, and it has additional COH. What is glutamine? It is something like an amino group present attached to the COH. So the net charge is actually 0. Otherwise, glutamic acid is negatively charged. It's an acidic amino acid. So what happens to this particular one, where glutamic acid is added to tRNA that is specific for glutamine? A second enzyme aminates it. Amination is addition of an amino group, something like this, so that now glutamic acid is aminated, then it becomes glutamine. Now there is no problem. We would get, by the action of the second enzyme which aminates this, you will have the conversion of glutamic tRNA for glutamine is now aminated to form glutamine tRNA for glutamine. Okay, I will stop here. Sorry for the interruption that we had in between, but I think we'll make up anyway. Yes, in the next class, we will discuss more about the tRNA, more on the translation, and then we will finish a few things. Yes. Please read the textbook. For this lesson, we might go into fair amount of details. So it's a good idea if you read the textbook as well. But otherwise, I'll stop here if you have anything to say. How is everyone? I hope you're all safe and fine. We only have 13 people attending, maybe 12. Okay, I'll meet you in the next class. Take care and have fun. And I hope you're doing the assignment, the final assignment, and then we'll calculate your internal marks. Take care and have fun.
Thank you.
Yes, bye bye.
Thank you, sir.
Thank you, sir.
Have fun.
Thank you, sir.